Good morning, ladies and gentlemen. It's a pleasure to welcome you to this session that is going to be discussing matters relating to COVID-19, particularly addressing the challenges and the current dynamics of the epidemiology of the severe acute respiratory syndrome corona 2 virus, and particularly addressing the challenges related to the variants and those that have been specified as variants of concern. You will realize that the current pandemic has caused a lot of suffering. Up to now, we have almost 181 million cases that have been reported. And of these, we have had almost coming to 4 million deaths globally. In Uganda, we have about 78,000 cases right now. We are in the middle of our second wave, and we have realized about 900 cases of death, and these continue to increase. Since the start of the pandemic, there have been significant efforts towards sequencing of these viruses. Right now, there are millions of sequences that have been shared within the public domain. And this has enabled the understanding of these viruses, but also enabled the design of vaccines and other measures to be able to control the disease. The impact of these variants and mutations that have occurred have had a significant impact on the epidemiology of the disease. These have had effects on the transmission of the disease globally and also between individuals. There has been an impact on disease severity, including mortality and morbidity. There has been an impact on vaccination as well as immunoprotection of the current vaccines and also those in design. But we have also recently seen a little bit of a difference because of these viruses on the ages that are affected by the virus. Originally, we thought that it was the elderly and those that were having other comorbidities. We see other age groups that were not initially affected getting affected as well. What is interesting is that there have also been reports of differences in symptoms because of these variations in the different viruses. And these have had a significant impact because people had or were used to certain symptoms. Because, for example, the current Delta strains don't seem to have a significant symptoms related to loss of smell. And therefore, people who rely on that kind of symptom have challenges thinking that they have either a normal flu or otherwise. So today we have two prominent scientists that are going to be discussing these challenges and evolution of this virus and bringing us up to speed regarding the evolution of this virus in Africa, and particularly focusing on Uganda and the variants that we see in Uganda. These variants have been categorized in what we call the variants of concern and also those variants of interest. And these eminent scientists are going to be describing these variants and their impact. So today we have two speakers, and I'm glad to introduce the first speaker, who is Professor Joloba. Professor Joloba is known to most of you or most of us. He's a renowned scientist. He's currently the dean at of the School of Biomedical Sciences at the Makerere University College of Health Sciences. He's very well known in the area of tuberculosis. He has established his career in tuberculosis. And when COVID came, he was able to repurpose his capabilities and the infrastructure he has built over time to be able to address the challenges of COVID-19. I will be introducing the other speaker, Professor Kalebu, when he comes in before his presentation. And allow me now to give the opportunity to Professor Joloba to tell us about the sequencing of these variants of COVID-19 and how this has impacted the transmission and other aspects of the disease within Uganda and within the African region. Professor Joloba, you are most welcome.
Thank you, Dennis. I hope people can hear me and see me. Okay, I welcome you all colleagues to today's World Health Summit. I'm very glad you could join both virtually and physically. So I appreciate the organizers for allowing us this opportunity to talk about this very important topic. My colleague Dennis has already introduced the magnitude of COVID-19, which is caused by the severe acute respiratory syndrome coronavirus 2, which is part of the other coronaviruses, which has really brought problems for human beings like SARS-CoV and MERS. SARS-CoV-2, the COVID-19, which started towards the end of 2019 in China, has really caused us havoc and the current pandemic. But my emphasis today is to show you how technology has helped us to quickly handle this epidemic. If you compare this epidemic to the previous epidemic, like the Spanish flu, the scientists have been very able to deal with this epidemic. So you can remember shortly after the epidemic, the pandemic, the whole genome sequence of SARS-CoV-2 was well established and known. And this quickly helped people who were designing diagnostics. And as you can see, within a period of 1 year, we do have vaccine, not one, but very many. And of course, there are attempts to deal with therapeutics. This has been possible because of the advance in technology. As usual, Africa was lagging behind in many of these, but I'm also happy to report that we have made efforts to catch up. So as I tell you, very many countries in Africa, as we will hear, also Uganda, we have capability to sequence the virus, to analyze the sequences, and be able to detect variants. And this is what we keep emphasizing, that the more you know, the better you can control these epidemics or pandemics. So there is really need for us to invest capacity in equipment and human resource to be able to handle these pandemics. On that note, I want to introduce my colleague, whom we are going to share this part of my session, Dr. Gerard Mbowa, who is a graduate of Makerere University and Baylor College of Medicine, and is going to quickly tell us about the genomics of SARS-CoV-2, the sequences, and introduce us to this part of topic. Gerard, there you go. I can see his presentations are up.
Thank you, Prof. Moses, for the good introduction you have made. And thanks, Prof. Diarugaba, also for taking charge of this and coordinating the presentation at large. So looking at CoV, and we have already seen from the previous speakers, they have laid down the burden of the disease itself on the continent and also globally. So I will start by taking us through a brief into what the virus is at the genetic level. So looking at the screen, what you see is the structure of the virus itself. Like when it comes to classifying viruses, this one was classified after sequencing as a single-stranded RNA genome of virus that you can see at the right-hand corner of this screen. So it has about 13 genes that are functional. And when after sequencing its genome, what was discovered was they would leverage about 4 to 5 different genes for diagnostics. So this is how it looks. And the important genes for diagnostics include the M protein, or the membrane protein, designated M here, which you can see here. These are the tiny spikes through its membrane. And then we have the nuclear protein, which is designated as N here. And then we have the S, or the spike glycoprotein, which is designated S here. And then there's the envelope small membrane protein, designated E here. So these have been utilized in the diagnostics. So when it comes to laboratory diagnostics, many of the tools that are in current use target at least minimum 3. So these are the genes I wanted to show you that are being used in diagnostics. And the more you use, the better, as we shall see. When you use one and all of a sudden the test you are running in the lab finds one of these mutated as a true characteristic of any virus, then you may be able to miss out on detection even when the disease is there or when the virus is there in the sample. So normally all the diagnostic techniques or tools that are being used, maybe what I didn't say, it's real-time PCR or multiplex PCR that is being used to detect the presence of the virus in samples in the lab. So looking at the diagnostic targets like you have already introduced, these 3 or 4 genes have been targeted. So you can see the ORF1AB has been, it's one of the commonest that is included in the panel when they are developing these diagnostic tests. So any part of that is picked or targeted. And then we have the spike protein, it is also that size. You can look at the relative sizes of each of them, and then E and M and N. So that's how it is designed at the genetic level. And like what Prof. Moses has already indicated, the journey to sequencing was too swift in that within just a month or 2, its entire genome had been sequenced and uploaded in the databases. So this presentation has also indicated the variants of concern as one of the challenges that has come up ever since we got SARS-CoV hitting the entire globe. So eventually, and then after a few months, about 6 months, scientists in UK realized that they are using a multiplex PCR, and they realized that one of the genes they are trying to amplify, that was the S gene, was not amplifying, while the other 2 were being amplified. So that wasn't something that was easily going to be detected and left unanswered too. So later on in the next few months, they realized that was a variant or a mutation had occurred in the S protein, and it was not amplifying in the routine diagnostic test. So here you can see the importance of genome sequencing. So in any diagnostic setting, if you realize you are not detecting any of those genes, then there's reason to understand that probably you have mutations that are occurring, and this is true to almost all the viruses. They naturally mutate. So I wanted to take you onto the continent and see how far the variants of concern have come. So you can see 30 countries in Africa out of the 56 have reported the alpha variant. And then when it comes to the beta, which was first discovered in South Africa, we have 29 countries reporting. And then when it comes to delta, which was first discovered in India, we have 13 countries. This list keeps growing every day. So there's a portal where this data gets updated on a daily basis. So we also talk about the whole genome sequencing. Its role has been immense when it comes to understanding the consequences of COVID itself, leave alone when it unveiled the opportunity to have quick diagnostics. It has enabled people to identify which variants are cropping up in different parts of the world. So whole genome sequencing briefly is the technique that enables someone to identify the entire nucleotide sequences of the genome or in the organism for this case. So that is what we call whole genome sequencing. So how we differentiate whole genome sequencing from targeted sequencing is, or for this case, what we do in routine diagnostics, which is real-time PCR, detects specific portions of the gene. But whole genome sequencing comes in to give you the entire genome, to give you the sequence from the start to the end. As you remember the cartoon I showed you earlier. So the whole genome sequencing will give you the entire span of the genome, and you're able to detect which regions have changed. Unlike the routine PCR, which just targets portions of that genome to just enable diagnostics or detection of presence or absence of the virus. So when it comes to analysis, that is one thing that when it came to Africa, that was a challenge. And if you remember in the introduction, Professor Biarogaba indicated that Prof. Joloba, like many of the scientists we know, no one had worked on SARS. But when the disease hit the globe, people had to repurpose their expertise. So that is what happened. And at Mulago in the College of Health Sciences, Makerere University, there was a sequencing platform and there were a number of real-time PCR machines. So after identifying the targets for detection in SARS-CoV-2, many scientists moved on to try to now purchase the reagents that are needed to do the detection. And at the moment in Uganda, we have more than one lab that are now doing detection. But when it comes to sequencing, due to the challenges associated with sequencing of the genomes themselves, very few labs are doing sequencing. At the moment in Uganda, we only have the one at Makerere University, the College of Health Sciences, and UVRI, as we shall see from Professor Pontiano in his presentation. So those are the only two facilities that are doing the sequencing. But why don't we have so many that are doing sequencing, yet we have a number of them that are doing the detection? So you have over 10 places that are doing the detection, but when it comes to sequencing, this is a highly specialized kind of process or technique that requires a highly specialized laboratory. So the challenges that are associated with sequencing of the genomes themselves include the cost of the reagents. Reagents are very, very expensive when it comes to sequencing. And there was a time, if you remember, some countries had put a blockade on shipping of reagents that's associated with the research or diagnostics of SARS. So there was that time also when it came to the severe phase of the disease last year. So that was one of the challenges that sequencing faced when it came to SARS-CoV. Then also the whole genome sequencing platforms are pretty expensive. Like when it comes to one of the sequencing companies, Illumina, its smallest gadget is $100,000 US. That's the Illumina MySeq. That's the smallest version of the sequencing platforms that can do sequencing of these genomes. So you can see you need a huge investment into procuring these platforms. Then also the other challenge is the analytics. It requires a lot of sophisticated kind of analytics that was not, I can't say it wasn't available. It was available, but it wasn't, people needed time to understand what they had to do when it came to this novel disease or novel pathogen. Then also the human resource, which also intersects with the WG, the whole genome sequencing, because you need people who are trained into whole genome sequencing and also people who can understand analytics itself. So that is the other challenge that whole genome sequencing faces. And then lastly is the commitment to fund this undertaking. So you realize that after even investing into the platform itself, you have to invest into either manufacturing the reagents, which is next to impossible, because the manufacturers of the platforms themselves are the ones that have this right to keep supplying you the reagents. So the commitment to fund the different aspects of this endeavor is also one of the challenges that keeps whole genome sequencing limited to just 2 labs in Uganda for this case. So what happened was the African Union had set up a specialized unit called Africa CDC. And that unit had started lobbying to overcome these challenges we have listed above. And one of their output is what you saw on the screen. They have a dashboard that charts different variants of concern across Africa. And they update this on a weekly basis. So this is what you are seeing here. So the total we have is about 7,000 beta variants. That's a South African variant. It's top on that list. And then followed by the UK variant at a total of 1,301 variants of alpha lineage. And then followed by the Indian lineage or the Delta. So that is 128 variants that have been detected. So, but even then, like we have seen, many of the techniques were building on already existing platforms. So about in 2014, they had set up a database that was called GISAIL, or the Global Initiative on Sharing of All Influenza Data. So this platform has enabled sharing of all influenza data. So it had been set up earlier and it was operating. So at the moment, it has 2 million SARS-CoV genomes, 2.1 million SARS-CoV genomes from all over the world. And we are going to see how many of those are from Africa. So out of the 2.1 million from that database, where every scientist or every sequencing facility is depositing those sequences, we have 2.1 from the entire world, but take note that we have more than 180 million cases that have been infected. So those will be equivalent to the number of genomes we would expect here. However, only 2 million have been sequenced and uploaded here. So you can see a big difference. So, however, from Africa, we only have 23,000 genomes coming in from Africa. And then on the sad note, we have countries that have submitted not any single genome from Africa. And the list is here on the right-hand side of the presentation. So we are lucky in Uganda, we haven't been part of this list. And on average, when you check that database, on average, you have 220 SARS genomes coming in from Africa every week. And then, like we see, 1% is from Africa. And those countries that have not yet submitted, there are efforts that are being undertaken to make sure they also start sequencing. So Africa CDC has set up specialized facilities where these countries are encouraged to ship their samples for sequencing. And the target by the end of this year is 50,000 whole genomes from Africa. So you can see we are somehow almost not even halfway there. So when it comes to Uganda here, we are not seated. There is 499 as of today morning genomes, SARS genomes from Uganda that have been uploaded here. And they have been uploaded by the lab at Makerere University, the molecular biology lab, headed by Professor Moses Joloba, and also the lab at Entebbe, headed by Professor Potiano. So when you try to break down how many of those in Uganda are alpha variants or lineage, so you can see we have 17 out of the 499 that have been uploaded from Uganda, 17 of those are alpha lineage. And you can see what makes alpha very important here. It was first detected in the UK, and studies show it spreads easily. However, we got to the other lineages. We have the beta. It has also been detected in Uganda. You see we have, out of the 499, we have 13 from the beta lineage in Uganda. And then still also, what makes these variants of concern unique is they are easily spread. And then out of the 499, the Delta variant, you see it tops the list. We have 38 out of the 499 genomes. 38 are coming from the Delta. So you can see where we stand when it comes to the distribution of these variants of concern. So we try to chart and see how the trend has been when it comes to sequencing these variants of concern. So there was, of course, they were first detected in the UK around the mid last year, but when it comes to the alpha, that was first detected in the UK. In Africa, it was first detected around January this year, and you can see how the trend has been. At the moment, we have more than 1,000 alpha variants of concern being detected in Africa. When it comes to beta, which was first detected in South Africa around December last year, you can see also the trend, it is picking up. And then when it comes to Delta, which was first detected in India, still we have it detected in Africa, and the trend shows they are increasing. So how do we start as a continent? Here I try to pull out the distribution of the number of COVID-19 cases. So you can see Europe has 47 million infected individuals versus the number of sequences that have been sequenced out of the 47 million is 1 million. And that contributes the percentage of 2.5 in the GISAIL. That is a database that is aggregating all the genomes of SARS that have been sequenced. So you can see how we are doing. Africa is in position number 3 when it comes to how many samples have we sequenced and contributed to this database. And then, so we have the Oceania, that is New Zealand and Australia, followed by Europe, Then we have North America number 3, Africa is number 4, then Asia and South America is the last when it comes to their contribution in this portal. So I think that is what I had for this presentation, but I conclude by trying to show what the Africa CDC has done when it comes to changing this picture, the picture we are seeing here. So Africa CDC got funding from Bill and Melinda Gates Foundation last year in October, and it has been funding specialized labs in Africa to try to pick up their sequencing so that they can contribute to this initiative. And we have to appreciate that without sequencing, then we will not be able to know if we have other variants that are popping up from different parts of Africa. These variants are of interest because, as we shall see in Prof. Pontiano's presentation, they have consequences that are beyond just being detected and documented. So I think that's what I had for this presentation. Thank you, and over to you, Chair.
Thank you very much, Gerald. Professor Joloba. Professor Joloba?
Yes, Dennis. I think now we hand it over back to you for our colleagues, unless you have questions for us or we get it later. It's according to the way you arrange it. Thank you very much.
Thank you very much, Professor Joloba and colleagues, and thank you, Gerard, for the very good presentation, highlighting the extent to which the African continent has come up to sequence their genomes and provide an overview of the variants that are circulating within the continent. That leads us into the next session, where Professor Joloba Professor Pontiano will be talking about the impact of these variants. Professor Pontiano is director of the Uganda Virus Research Institute based in Entebbe, and he holds a medical degree from Makerere University, but is also a professor of immunovirology at the London School of Hygiene and Tropical Medicine. Professor Pontiano is very well known for his work in HIV and HIV vaccines. And just like Professor Joloba, when SARS came to Uganda, the UVRI repurposed their infrastructure and capabilities to be able to address the challenges of COVID-19. So I will hand over to Professor Kalebu to take us through the impacts of COVID-19 variants in Africa. Professor Kalebu. Professor Kalebu.
Chair, Professor is joined as an attendee. So let me have IT upgrade him to a speaker. He may not be able to speak as an attendee.
Thank you. Please do that. So while Professor Pontiano comes, here he is. Professor Kalebu, you are most welcome. So please proceed. I introduced you in your absence. I hope you were able to hear me. So I suggest you go straight to your presentation so that you can give us an overview of the impact of COVID-19 variants in Africa as they relate to several aspects, including transmission, including diagnostics and vaccination, and several other impacts that the audience will be very much happy to listen to. Professor Kalebu, you're welcome.
Thank you very much. Can you hear me?
Yes, we do very well.
Okay, and I think let me also see that you see me. So apologies, I had some issues with the connection, but let me start first of all thanking the organizers for inviting me to give this presentation. Unfortunately, I missed the earlier speakers, and hence there could be some repetition of what I'm going to say, what other colleagues have already talked about. But in my presentation, I'll talk about the genetic composition and structure and the relevance to virus phenotype and the way the virus functions, the changing sequence patterns globally, Africa and in Uganda, and the known impact of these variants and the evolving picture in Africa. And then I'll go to the conclusion. This is just a reminder of the large virus that we're dealing with, RNA virus, with a structure that looks like that, hence the name, with a genome, a large genome of about 330 kilobase and single-stranded. I think for the audience, all I want to take note is, of course, this is a large virus. It has a number of regions that are important. It has the spike region, which we'll talk a lot about, relevant to vaccines and disease and transmission, the nucleocapsid protein, the envelope, and the membrane glycoprotein, and this single-stranded RNA. And this is down in there, the cartoon that shows some of the genome of the virus, with the open reading frames and the other structured proteins, which genes that lead to the structured proteins I've talked about, the spike, the envelope, and so on. The virus mutates as it replicates, like many viruses, and the replication is random. The errors made, bases changing, also to create substitutions, but also it can have deletions and insertions. Some of these base changes lead to protein changes, others don't. And for SARS-CoV-2, it is thought that you have 1 in 1,000 substitution per site per year. And that translates into 1 substitution every about 11 days. It's not comparable to HIV, which changes much more than SARS-CoV-2, as shown here. At the start of the pandemic, 2 lineages were described, lineage A and lineage B. And these came at around the same time when the virus was described in Wuhan as those 2 lineages. And their lineages did spread very quickly all over the world. And this slide is of December 2020, but it shows how the different A and B lineages spread quickly all over the world within a very short time after the first description of this virus in Wuhan in China. Currently, there are a number of sequences, and I'm sure probably colleagues who have spoken before have mentioned this. There's a lot of effort to sequence the virus. Currently, we have by May, we had over 400,000 genomic sequences that had been generated through different epidemiological studies across the world. And these have been deposited in the GISAIL database. And by that time, Africa had sequenced about 14,500 sequences. And the majority of these are from South Africa, nearly half of these. Of course, some of these sequences do not meet the quality that is required. They have some gaps, but you can infer and understand still the same, the pandemic. This slide shows a bit of what has been done in Africa, the sequences that have been generated in Africa. It's a bit old. It was in May 2021, and it's part of a publication that is under review in Science, of which we are part, looking at a year of genomic surveillance, and that shows the way the pandemic has unfolded. And from this study and this work that is being reviewed, it's clear that in Africa, a lot of the early viruses were introduced to the continent between February 2020 and February 2021. And we had many different introductions from different sources. And at least using the sequences, phylogeography, phylogeny, you can infer how these viruses were transmitted. You can see some countries don't have any, like South Sudan here, South Sudan, but we are currently sequencing also for South Sudan through WHO. I want to talk more about the variants of concern. When, of course, it was noted that the virus was changing. Towards the end of last year, it was shown that there was a new strain or a variant that had many mutations, more than 20 mutations, that was of concern in that it was transmitting a lot in the UK and was called the UK variant. The first one that was described, the UK variant, now called the Alpha variant. And then other variants were described that were of concern. And when we say variants of concern, these are associated with or more of the following changes at the degree of public health significance: increase in transmissibility, or detrimental change in COVID-19 epidemiology, or increase in virulence of the virus, or changes in the clinical disease or presentation, or disease that can affect other public and social measures available, like diagnostics, vaccines, and therapeutics. And many mutations we are seeing in these variants of concern. More than 20, but most important are the mutations, the spike region, which is important for the attachment of to the S2 receptor, the human receptor, and hence these mutations leading to increased attachment, better attachment, and better structure for the virus to attach better to the human cell and to transmit better. So far, these are the variants of concern that have been described. These are 4. I've noted here the mutations. There are other mutations, but some of the concerning mutations in the spike is the 4501 for the UK variant, now called the Alpha, that leads to increased transmissibility. And there have been some indication that it could also increase on the severity of the disease, leading to increased hospitalization and death. The South African variant, which also has that mutation in the spike region, but also the E484, which again improves on the structure of the spike and hence increasing on the chances of attachment and transmission. The South African variant is now called the Beta variant. It has been shown to have increased transmissibility, some increase in disease severity, although that is not so clear, but indeed that has been also described. When you use some of the neutralizing antibodies, monoclonal antibodies, it is more resistant to some of these antibodies. And as far as vaccines are concerned, there have been a small study that has indicated that it may not prevent mild disease and moderate disease, but it's some of the vaccines like AstraZeneca, of course, can prevent severe disease, but it has some effect on some of the vaccines. The P1 from Brazil, which again has those mutations, 501 and E484, now called the Gamma, it has been shown again to have increased transmissibility. It has some effect on the neutralizing antibodies, in that some of the antibodies are the virus is less sensitive to them. Then finally, the more recent one, the Indian variant, now the Delta variant, which again has increased transmission rates, even affecting the young population. There's some evidence, although it still needs to be proven, that can also lead to more severe disease, and also it has some effect on neutralization. And for the vaccine efficacy, no data is available as yet, but more information is being collected. Transmission is an important factor. At first, when the Alpha variant or the UK variant was described towards the end of last year, it was increasing transmission in the UK. And at that time, it was described as being 50% using mathematical modeling and other approaches, 50% more transmission than the one strain. But you can look at the Delta. Delta is even 60% more transmissible than the Alpha. And this is what is happening. Since the Delta was described not long ago in India, it has now been detected in 85 countries, including several African countries, and it is rapidly becoming dominant in many countries. This slide again emphasizes, I know it is small, but again, it's a good publication that came out in the New England Journal of Medicine, a brief publication by Slim Abdul Kalim and Turio in South Africa, again, looking at these strains, variants, and also the efficacy as far as vaccines is concerned. I must say, when we talk about vaccines, there's work that has been done in the lab to look at neutralization, but more needs to be done to look at the real efficacy, how these vaccines affect when individuals are infected with these strains. As you can see, some of these strains are affected by some of the vaccines, like the Pfizer. Neutralization is reduced in the lab, but no efficacy data. When you look at the Sputnik of Russia, again, you don't have a lot of data. AstraZeneca, at least what we know, as I mentioned with AstraZeneca, is the reduced effect in a small study of mild and moderate disease, but we're still confident that the AstraZeneca will be effective as a vaccine against severe disease. But more work needs to be done. I'll refer you to this publication for some more details and also references that are given. There are also variants of interest. Again, these are called variants of interest because when you compare them with reference strains, they have mutations that have been established that are suspected to have implications on the phenotype of the virus. And these have also been observed to cause community transmission and multiple cases and clusters. And they've been detected in multiple countries. All, in other ways, they have been assessed to be variants of interest by WHO in consultation with WHO SARS-CoV-2 virus evolution working group. I must say the grouping of some of these viruses also differs on which group. The WHO has a way of grouping them, the CDC has a way of grouping them, the Public Health England has a way of grouping them. Generally, it's the same, but there are some minor differences. And listed here are the strains that are called the variants of interest. I won't go into the details, but I want you to look at the last one, A23.1. This is not listed, but it has a mutation that has been classified by the Public Health England, the E484K mutation, to be a variant of concern, of interest. And I'll talk a little bit about this later in my presentation. Finally, the variants of high consequences. These cause more severe disease, greater numbers of hospitalization, and it has been shown to defeat medical countermeasures such as vaccines, antivirals, monoclonal antibodies, and so far none of the variants meet this definition. But this is a category that has also been described as one of those variants of high consequence. This slide, I wanted to show you the changing pattern in Africa. Again, this is part of the publication that I mentioned that is being reviewed now, that is looking at this changing pattern of these variants. At the beginning of the pandemic in Africa, in March, May, you could see different viruses and sequences of viruses from different parts, and these were matching where these individuals were coming from. If somebody came from the UK or USA, the viruses in Africa were matching those. But as we moved into January and March this year, you can see in Africa generally a dominance of the alpha and the beta. The alpha, the UK strain, and the beta, the South African strain. That is up to March. In South Africa, they have seen 3 waves. In fact, currently they are in their 3rd wave. They had the first wave in June, July, August, the second wave in November, December, and now in May, they are beginning to see they're in their 3rd wave. And I want to see what are they saying in the terms of the variants they have. In the second wave, there was a dominance of the beta variant, the South African variant, but now in some parts of South Africa, they are beginning to see an increase of the Delta. The Delta that was just recently described in India is now becoming a major variant in South Africa and a major contribution to the 3rd wave that they are having. Coming to Uganda. Uganda, I think we have had 2 major waves. The first one was in November, December, towards the beginning of January, And now, beginning of May, June, we're in our second wave, and everyone is aware of this. What's happening in terms of variants? When we first started sequencing here in April, and we looked at variants from March, since when the viruses were introduced, we started also seeing at the beginning variants or sequences that were matching where individuals were mostly coming from. Whether if somebody came from UK, they matched those in UK, South Africa, or in other places in the Middle East. Now, in June, July, we started seeing another variant, the A23, that was seen in a cluster of infections in a prison in Amur, in Kitgum. It was called the A23, and that was dominant in that region at that time. But as time moved on to November, December, it appears this virus evolved or mutated to the strain I mentioned about which is a variant of interest into the F23.1. And that strain started becoming dominant in our sequences. And by the end of the year, December to January, early this year, it was dominant in the sequences that we had from Uganda, especially from Kampala. So this was a strain that caused us some concern. And this has been published in Nature Microbiology for those who want to see more details. Now, what's happening as we see in the second wave? We say now the positive rates in the second wave, in the first wave, was about 750 positive per day in the first wave. As I said, in the second wave, we have been sequencing about 20 to 40 genomes per month. In the first wave, as I mentioned, we saw that strain of A23.1, which was dominant at that time. Yeah. Now we are seeing a dominance of the Delta, the B1.617.2. That strain that I've mentioned that is of concern, transmits very easily, and has other characteristics. This is what we're seeing, preliminary data, but we need to look more. In the last sequences that we had, out of 31, 30 were of Delta, samples that collected from Kampala and the surrounding areas. Now, could this explain the differences we're seeing? Clinicians have been telling me how they're seeing younger people coming up with severe and critical disease that we did not see in the first wave. Could this variant be the cause? We need to look more. But indeed, we are seeing a changed picture of our clinical COVID. Of course, we're also seeing more deaths, but this could be because of more transmissions. But we need to look more. Is Delta also, of course, affecting the picture and the severity of disease in our country? As I end, what needs to be done? I think in Africa, we need to do better. I've said only 1 out of 300 sequences in the GISAIL are from Africa. So we need to do better. We need to look more into high and low transmission areas. We need to look in those who have been vaccinated versus non-vaccinees. We need to look in those who have severe disease. With the aim, we have been discussing with our groups, the epidemiologists, those in the lab, clinicians, how much should we sequence? Some people are saying we should sequence about 30% of the positives. It's a lot of work. It's quite expensive to sequence. It takes a lot of time. We need resources to do that. But we need to do that so that we can understand more the impact of these variants. Protocols are being developed in many countries in Africa. Some are ongoing and others are planned so that we can understand, especially now as we roll out vaccines. So in conclusion, I can say, although we are looking and following the pattern in Africa, there's limited sequencing in Africa and we need to do more. I've shown you that we have a changing pattern of variants. Now we have a dominance of the variants of concern and the variants of interest in Africa. And the variants of concern are associated with the waves we're seeing, at least in South Africa and possibly in Uganda. But we need to investigate more the impact. I'd like to end by acknowledging those who provided me with some of the slides and those who have provided funding for the sequencing, the patients and the staff of UVRI, the MRC UVRI London School, and the Center of Public Health. Thank you.
Thank you very much, Professor Kalebu. That was quite a very good summary of the current situation regarding the variants in Uganda, and particularly the impact on transmission, the impact on vaccination, and the impact on severity of the disease, particularly in the different age groups. I want to open up this session for discussions, and I will ask the organizers to allow the attendees to ask questions so that we can get into this discussion. While we do this, Professor Karibu, I have a few questions for you. You alluded to the impact of these variants on vaccinations. And as you very well know, there is quite a lot of concern regarding the effect of the vaccines themselves. Very many people are worried about taking their shots because they think that a few people have died, which is correct, that people have taken shots, and a few of them have died, which is possible. So I would like you to highlight a few impacts of these Delta variants with regard to the AstraZeneca vaccine that is currently being used in the country, and vis-a-vis some of these deaths that might have occurred in people that either have been partially vaccinated or have been fully vaccinated, so that we can create confidence within the public that probably the vaccines still work. So if you can comment on that, please.
Okay. Let me talk as a scientist. As a scientist, when I want to find out a conclusion, what something does, I have to design my study very well and look at the sample size and the numbers. I know, and it's unfortunate that people have died who have passed away after being vaccinated. Yeah? But you need to go in details and see how many are those who have died after vaccination and how many have died without vaccination. What are the numbers? That has to be clear. And also those who have got vaccinated. At what stage were they vaccinated? You know, we're vaccinating would be for testing. Were these individuals infected during the time of vaccination, immediately after vaccination? We know that the immune response for AstraZeneca after the first dose comes after about 3 weeks. And after then it's lower, then you boost after 9 to 12 weeks. And the immune response is even better 2 weeks after the boost. And that's when you have maximum protection. So when do these people get infected in relation to the first dose and the second dose? All this information is to be done. And that is the reason that we need to do very good studies. And I've said these studies are lacking, but we need to do them now so that we can address some of these issues that our colleagues, the public is asking, so that we can respond in a very, very affirmative way after scientific basis. I can tell you what we are seeing here. Within our staff, it's preliminary and we want to do more. We have looked at individuals who have been severe disease, hospitalized, moderate and mild disease. What we are seeing are pictures, at least in the few staff we're looking at and want to do it more, is that individuals, most of our staff who have been in hospital, who have had severe disease are those who didn't get vaccine. Those who got 1 dose, we have had some who have had some mild disease, and fewer infections, more infections, those who didn't get the vaccine, and the least number of infections are those who got the 2 doses. So that gives us some indication. But best, because of that, in Uganda, we don't have a lot of information. Let's look elsewhere. Elsewhere, AstraZeneca has been given to millions. And what we're getting is that AstraZeneca has been of benefit in countries where they have vaccinated many people. And the concerns of causing death has not come up. I want to assure people that the vaccine is safe, but we need definitely to look more so that we can provide better scientific explanation. But getting 1 or 2 individuals here and there may not give us the right answer. I hope I have tried to answer your question.
Yes, thank you very much, Professor Karibu. Indeed, there were 2 other questions that were related to this, and you have ably answered them. People are very, very worried about these vaccines, and thank you very much for allaying their fears, and particularly to allay their issues related to safety, but also to protection. That as far as the data available is concerned, is a high level of protection even against the current circulating variants. But of course, there are those other additional deaths that have occurred in which further investigations may need to be done. There's another question about the different variants, whether they require different treatment approaches and if it is possible to do this. Could you address this? Thank you.
Yes, and I think other colleagues will also come on. One thing I didn't mention is diagnostics, because again, that is a word about diagnostics. Indeed, as viruses change, we need to be aware that that can affect diagnostics, especially PCR that uses primers that are matched to the sequence of the virus. The good thing that a lot of our diagnostics are not only targeting the spike region, but they target other regions. They touch envelope, nucleocapsid, and many others. So we have more than one target. But indeed, we need to continuously look at our diagnostics and primers so that we match the changing viruses. And for that reason, we, as I chair the quality assurance committee for the lab, we shall be ensuring that we have panels that would send to different labs. And in these panels, and colleagues have made up these panels, we will include samples from these variants, including the alpha to make sure it's picked, the delta and the rest. But so far, I can assure the public that diagnostics is still okay, but we need to work more. As far as treatment is concerned, there has not been any description that you will change management of the patient depending on the variant that you have. It could cause severe disease. They could cause diseases in the young and different age group, but the management remains the same. If anything changes, or if there's anything that I'm not aware of, as you know, COVID, we cannot say we know everything, because every time if you don't read, you may miss out on some important information. But what I'm giving is as far as I know, but doesn't mean that it will not change. Treatment remains the same.
Thank you very much. I want to throw this at Professor Chirenga, who is a panelist here and has been at the helm of COVID treatment in Mulago. And he might offer some bit of experiences that they have seen throughout the period that we had the earlier variants of A.23.1, but now with the Delta strain, if there's any recommendations for changing treatment. Dr. Chirenga, please.
Thank you very much, Chair. I think what Professor Caleb has said is what is known now, that probably the treatment of COVID is not yet affected by which variant is causing it. But of course, when we get to the phase where we have antivirals, then this discussion will come up. Right now, actually, when we tell you we are treating COVID, we are actually not treating it. We are just fighting around what it is doing with the inflammation, with the respiratory failure. So we are not treating it. When we get to the stage of antivirals, this discussion will become important. Thank you.
Thank you very much, Dr. Chirenga. Are there any other questions from the audience? In the meantime, Dr. Chirenga, probably we may also take this opportunity to talk a little bit more about the treatment options. As the virus has mutated, so has the mutation of the health-seeking behavior of the population. When this Delta, at the time actually this Delta strain came, of course they had no knowledge of what was happening, but there was a very serious threat that everybody was rushing to buy antibiotics, buy vitamin C's, buy zinc. Could you please take this opportunity to probably pass a warning, but also some additional information to the listeners on what this treatment for COVID-19 is and what the public should not be doing?
Okay, thank you again for bringing that up. Oh, I can, maybe let me show my video. So what, the treatment for COVID has been also been mutating, as you suggest. At the beginning of the pandemic, there are many things which we tried, which science has since shown they have no benefit. Now, those are very many. Starting from, I would start with some drugs like hydroxychloroquine, which was used a lot, convalescent plasma. All these treatment modalities have been so far found. Antiretroviral drugs, Pinava, all these have now been shown that they offer no benefit.
Ivermectin?
Ivermectin, yes, but that one is still hot. I had not wanted to include it in this group because I still think it belongs in the group where we need to do more research. But there are those ones where we have good clinical trials and we have concluded the matter. Hydroxychloroquine, anti-HIV drugs, convalescent plasma, those ones we have concluded. Ivermectin is still sitting in that middle group where we need more evidence of its effectiveness. Now, going to the ones which are used in home care, for example, vitamin C, zinc, these we use them a lot in the beginning. Now, our change in these immune boosters has been on the severity of the disease. If people have early disease, they can take zinc, they can take vitamin C, they can take their fruits, all these things to boost their immunity. However, what we see is that people who have severe disease, there is no more role of giving them vitamin C. You are just going to cause them more gastritis and cause problems. So we discourage those ones. Now, what has been happening is using these things in high doses. I actually know a colleague who was taking 15 tablets of vitamin C per day for like 3 months, and he came down vomiting blood. And this is actually what happened. So we need to encourage people to use, but under prescription. Now, the other thing which comes up among the new things is vitamin D. The evidence is still weak, but you have seen it has cropped into our guidelines, and people are using vitamin D. So, Chair, maybe to conclude is that the treatment is mutating as the virus mutates, but for it is mutating as our knowledge improves. Now, what is remaining useful? Maybe I need to tell the listeners that what is remaining useful are the steroids, notably dexamethasone. But as you know, in the clinic, we don't stick only to dexamethasone. We can use any other steroids, provided we have balanced the doses, whatever is available. Anticoagulants, to blood thinners like Clexane, anti-IL-6 like tocilizumab is now concluded by the recovery trial to be useful. So those are the 3 things that we have now that are known to benefit people who have COVID.
Thank you very much, Professor Chirenga. I think that was very, very useful. The public is certainly very anxious. And you can switch off your video. I can see you are a little bit stressed, but we understand. So thank you very much. I think one of the pertinent questions, and I think I need to throw this to Professor Kalebu, is that vaccination has been going on for quite a while now. Do we know, or have we ascertained the level of protection within the population, either as a result of natural infection or as a result of vaccinations? Not just determining that people have seroconverted, but the levels of protective antibodies within these individuals, and particularly those who have been sick or those who have died. I don't know if this kind of information is available, and if it would be very useful, so that it can also reassure the public that actually the vaccines are working and actually are inducing protective immune responses. Professor Kalebu, do you like to have a go at this?
Are you talking about Uganda in particular or globally? Yeah.
Uganda, if there's any data.
Oh, Uganda, we're still behind. A lot of protocols are being developed very quickly. First of all, we are collecting samples, individuals who have been naturally infected, to look at the natural immune profile. How long do the immune responses last? How high are the immune responses, both in terms of humoral T cells? Studies are going on to collect samples, but also studies among vaccinees. Unfortunately, for now, we don't have any data. But that's an important question. I believe it will not be any different from other places. But as you know, based on some other vaccines, vaccine responses could also differ based on individuals' host immune responses, both based on the environment. So I think what you're asking is an important question that we need to look at. Let's not assume that what we see elsewhere in the North will be the same. So we don't have an answer now, but studies are beginning, are ongoing. Hopefully we shall be able to give you the immune responses in vaccinees and how protective these vaccines are. Thank you.
Thank you again. Unfortunately, there's so many questions are coming to you. One of the other questions is the current observation of the infection in the middle age groups. But the earlier recommendation for vaccination was for those who had comorbidities and those who are elderly. Apparently, the government hasn't come up as yet about the recommendation to vaccinate these now highly vulnerable individuals, but also the children. We have seen some children also who have come down with the disease. And unfortunately, some of these vaccines haven't completed trials in these age groups. Can you comment about that, please? Thank you.
I think, although we have said the picture is changing, we have young people getting infected, still the high-risk groups are the elderly and those who have comorbidities. They are still, the way the government and the planners describe all the age group, comorbidities, healthcare workers at risk, they are still at risk of getting the disease, but also having severe disease. We see the disease in the young population, yes, but I think those are not as high. But my suspicion, and this is what has happened in other countries where they have enough vaccines, if we have enough vaccines, then we should start going down to the lower age group. And as more studies are being done also to see the safety and immune responses in the lower age group. So indeed, if a young person had an opportunity to get a vaccine, he should take it. But with this shortage of vaccines, I think we see the priority has been with those individuals who are likely to have severe disease or are likely to get infected and all that. But with the availability of more vaccines, I'm sure the age groups will lower, just as has been done in other parts of the world.
Thank you very much, Professor Kalebu. Another question to Dr. Chirenga as we wind up. One of the impacts of these new variants is the difference in the symptoms. Some publications indicate that, for example, the Delta strain does not show some of the symptoms in the patient, such as loss of smell. What are you currently seeing in Mulago, and what would you want the public really to understand about these differences in the strains? Because people were used to loss of smell, those who use perfumes. If they lose the smell of their perfume, then they know they are finished and they rush for treatment. So Dr. Chirenga, would you want to comment about the typical symptoms that you see, particularly with this wave that is dominated by the Delta strain?
Thank you, Chair, again. First of all, I put a disclaimer that we don't have enough sequence data in our patients to be able to relate it to the symptoms. But like Professor Kalebu and Gerard showed earlier, some variants, what we know that transmissibility is increased, severity is still a question of debate. In terms of symptoms, I will tell you what peculiar thing we have seen with the current wave. You know, the COVID at a clinical level, we look at it has like two phenotypes. The phenotype, which is bronchitis, which involves the respiratory system. People have the normal flu, pneumonia that you know, cough, chest pain, all the respiratory symptoms. And then the phenotype, which is about 20 to 30% of people who are suffering from thrombosis. They only have clotting in their lungs, they desaturate, they have respiratory failure, but they don't have a respiratory phenotype. Now, what we have seen actually with the current wave is that people are coughing more. There's more cough. In the first wave of COVID-19, we admitted people who are very sick, but they didn't, you even wonder whether these are people with respiratory failure. They didn't have respiratory symptoms. But what we have seen in this wave is there is more cough, there is more chest pain. About loss of smell, I will have to look at my database. I don't think there is a big change, but the big change has been an increase in the number of people who have respiratory symptoms. And I think this is important because, as you know, when people cough, you generate aerosols, and that actually could explain the increased transmission that caught Kampala like a frame, because everyone was coughing, people were generating aerosols, meaning that there was more transmission. Now, we have now collected a series of patients who are being discharged. I think now we have reached around 270. So we'll be able to provide data, and this is an electronic database. Now, to take you back a little bit about vaccination, in this database of 260 people, we have 26 who have been vaccinated. And most of them had got 1 dose, and I think 6 had got 2 doses. So now with that data now, we can compare severity about whether you got 1 dose, whether you got 2, whether you did not have, and we'll have that information very soon. Thank you.
Thank you. Thank you very much. We have come to the end of this session, unless somebody has an extra burning question. I want to thank the presenters, Professor Joloba, about the genome and the current sequencing, and Professor Kalebu for ably giving the highlights of the impact of these variants, and Professor Dr. Chirenga for coming in with the current treatment that is going on in Mulago, and practically actually what these variants mean for people who are managing these patients. I want to thank the attendees. We have had a record attendance of about 47 individuals, and I want to thank you very much, and I wish to end here and hand over to management. Thank you very much, and wish you a very good conference for the rest of the days. Thank you.